Hello, everybody. Welcome to our first Engel rounds of 2024. And our presenter today is Paul Geha. And I'll introduce him more in a minute, but I just want to go over the usual housekeeping items. One is, if you're watching this on Zoom, your camera is off and your microphone is off. If you're here in person, of course, we can see you all and hear you. Also, if you're on Zoom, don't send messages or questions using the chat box. That's reserved for the panelists. But if you do have questions or comments, and we hope you do at the end, near the end, please use the Q&A box at the bottom, and then we'll get through as many of those questions as possible. Also, there'll be a QR code coming up at the end and a link for the evaluation, and we really hope everybody who's here or watching on Zoom completes the evaluation. And that takes care of the housekeeping items. So it's my pleasure today to introduce Paul Geha. Paul came to us from Yale a number of years ago and has given Engel rounds before. And it's really been amazing to see what's happened in the past few years since he's gotten so many new grants and started so many new projects, and his research program on the biological brain basis of pain and chronic pain has really taken off. So I'm really glad that he has a chance to present today and give us an update on what's happening in his lab.
Yes. Can you hear me? Okay, thank you, Steve, for the invitation and for the introduction. So today I'm going to give you an overview of what we are doing from the brain point of view on chronic pain and the brain. So from mechanisms to biomarkers. So I would like to start, though, by this news. I think many of you have seen the news. The Queen of Denmark, Queen Margaret II, announced that she was giving up the throne after 52 years. So I quote, with her voice cracking, she said, "I have decided that now is the right time. On 14th of January, 52 years after I succeeded my beloved father, I will step down as Queen of Denmark." In her speech, the 83-year-old Queen said that back surgery, actually in other references, they said extensive back surgery that she has done in February of 2023 had raised thoughts about the future for her. So Queen Margaret II was the longest serving monarch after the death of Queen Elizabeth II. And I put this up there because it caught my attention when I heard the news about how extensive the effect of pain could be, or chronic pain, on our motivation. Right, so to the point of giving up the throne. And that's what I will emphasize today in our view of how we look at chronic pain. We always are going to look at it in the context of motivations. It's always something that we need to factor in our decisions to stay at work, for example, or to leave because of the pain. And also in nature, as I show here in this picture, pain means different things for these different animals. So it's always in the context of motivation. So, and nowadays, I mean, there are lots of interest in the literature on shared mechanisms of chronic pain and emotional motivational problems, or the challenges of pain and fatigue, because they always come together, and clinicians in the audience would know that very well. However, what we have, or what has been the gold standard so far, is the numeric pain rating scale, for example, or the visual analog scale, or the FACES pain scale, or sometimes we can rely on well-validated questionnaires. But these probably won't capture what our patients are going through, and why would they do what Queen Margaret has done. So we therefore actually set to study anhedonia in chronic pain patients. And we set to study it in different ways, from the perceptual side and from the motivational side. So this is a recent work that we have started to look at to measure actually motivation in patients with chronic pain. So we brought them to the lab and we asked them to play a game, basically that where they are faced with an easy task and a hard task. The easy task, they have to achieve certain number of presses of the button in a certain time to win $1, but that's it. The hard tasks, it comes with different probabilities and different earnings between $1.5 to $3.5, but they have to use a harder way. So they have to basically press the button with the pinky finger of their non-dominant hand, and they have a shorter period of time to achieve that. So, and this was in collaboration with David Zal at Rutgers. We've tested actually non-depressed patients. So these are patients who have back pain, but they are not really clinically depressed. And we compared them to pain-free controls. And what we clearly see here is that chronic pain patients are less willing to expend efforts compared to the pain-free controls. The patients are shown in blue, and the healthy controls in green. And what we are plotting here are the choices, basically the hard choices. How many times, on average, these groups go for the hard choice at different probabilities of winnings, or different probabilities of different amounts of winnings, and different probabilities of winnings. And you can see clearly that there is a loss of motivation in this sense here to go for the hard tasks in the patients. And this doesn't happen in the context of a generalized cognitive decline, for example, because we have done in a subgroup of these patients, we have done tasks such as the Wisconsin card sorting task, which measures executive function. And these patients seem to be doing quite as well as the pain-free controls. So it's more or less specific to motivation. And in some of these patients, we have measures of their brain structure, T1 images. So we measured the brain volume of the area that we know very well is involved in motivation, the accumbens volume. And we see that the accumbens volume becomes tightly correlated with the choice, the high probability for high cost, high reward choice. So the hard choices in the patients only, not in the pain-free control. And I'll come back to that in a few slides. So what about perceptual anhedonia? So this was an older study that we have done, but I will start by data from, I think, Sweden, showing actually that if you just ask patients about their life in general, using the chronic, so using the Snaith Hamilton anhedonia scale, their subjective report of anhedonia shows clearly that they are worse than the healthy controls, so than pain-free controls, even though they are not, for example, misusers of drugs and alcohol. And they are also, so people who are misusers are worse than the people who are non-misusers. So chronic pain patients have anhedonia on self-report. This was a study that was done by Garland et al. In 2019. But before that, actually, we tested the perceptual anhedonia by presenting patients with food. So we brought a cohort of back pain patients who, again, were not depressed, and a cohort of pain-free controls. And they were basically matched on all measures. And then we asked them to taste different concentrations of fatty pudding with two flavors, the vanilla flavor and chocolate flavor. And then as a control also, we used sucrose solutions with increasing concentrations of sucrose. And so assuming that there will be an increase in liking in all of these stimulations. So in the first part of this experiment, we asked back pain patients to actually sample these foods without consuming them, just orosensory perception. And we used for liking this bipolar scale, which actually uses anchors to have the patient rate their experience of liking over a scale that should encompass all types of liking in their life. So it's called the labeled hedonic scale. And so this is what we observed. We also observed that similar to the task we've seen with effort expansion, that the patients show anhedonia. They are in gray here, and for increasing concentration of fat, they have less liking than the healthy controls. But this doesn't happen for sucrose, and we have a theory about that. I will mention it later. But it does clearly happen for high-fat pudding. And all of the other sensory perceptions, I don't have them here, but all of the other sensory perceptions of the food were normal in these patients, but their perception of pleasure basically was disrupted. And when we brought them back on a different day, we asked them to come hungry, actually, and we asked them to consume the pudding that they liked the most from the first session. And so the change in hunger was similar between pain-free controls and back pain patients. The pain-free controls are in black here. The calories consumed were actually the same, but the notable part was that the liking of the pudding from the day before or the first session predicted caloric intake only in the healthy controls. So liking in the patients was completely not unrelated to the caloric intake. They must have stopped for other reasons, of course, but there is a disruption now between liking and caloric intake. So there is a disruption in hedonics, and that disruption in hedonics could have an impact on how these patients would consume certain types of foods. And again here, we see that the liking was tightly related to the volume, again, of the accumbens. So in another different data set, completely, collected at a different site, actually, the liking is tightly related to the accumbens in the patients only, but not in the healthy control. And one preliminary conclusion we can make here is that hedonia in patients, or their experience of pleasure, becomes really tightly related to their limbic brain, to their accumbens in this case. So actually, there has been a lot of interest in interventions that would increase now or train patients to savor rewards. This is an editorial written by Eric Garland, where he advocates for basically new ways of targeting untapped therapeutic opportunity of increasing patients' ability to enjoy pleasure. So the first conclusion we have from this is that chronic pain is associated with measurable anhedonic changes. So these changes are in line with clinical observations and present additional and distinct dimensions to assess and treat beyond the pain intensity scale. So, but now we are actually interested more in mechanisms. And so where do we look now if we want to go through all the, if you want, nociceptive and then the cortical pathways that are involved in pain? So how do we look, or where do we look now in the brain? I gave you a hint, of course, from the previous slides with the accumbens, but there are so many other pathways involved in pain in the periphery and in the spinal cord. And how can we basically look at the brain to understand this behavior and explain it, and also from there move on to develop comprehensive and reproducible biomarkers that are quantitative and that would help us deal with this problem of assessing pain in a more comprehensive way and in a more valid way. So this is what we are faced with, though, when we look at the brain. So all of these areas that are colored in blue and in red will receive some kind of nociceptive input from the periphery, directly or indirectly. The ones in red are colored in red because they are most commonly seen when you do an acute painful stimulation in the scanner. But there are multiple pathways that travel through the spinal cord. Some of them actually do not synapse in the thalamus, so they don't go through the major sensory hub of the brain, but they go through the parabrachial nucleus in the brainstem and directly feed into the limbic brain. And so, like I mentioned before, the accumbens, and I mentioned motivation, and I mentioned liking. So this is what prompts us to also talk about the fact that learning circuitries are going to be involved in chronic pain, like brainstem, hypothalamus, etc., including striatum and cingulate cortex. These are brain areas that are important in memory formation and that are important in learning, and that receive a lot of them receive actually mesolimbic dopaminergic input. And this is something to keep in mind as we go through our findings. So this is a result from a large analysis of 4,160 cortical recordings that was done in Canada, trying to basically understand, again, how do we feel pain in the brain, right? And so only 1.4% of these, actually, these are stimulations, not recording. So 1.4% of these recordings were actually exclusively painful only. They elicited pain in the patients, and they were exclusively concentrated in this small area shown in red here in the posterior insula, secondary somatosensory area. So the conclusion is that from this paper is that it's very hard to find a pain cortex. So it's not like the visual cortex. It's not like touch. And the cells that are there, according to this paper, are sparse and very hard to detect. And they are an extreme minority of the cells that they were stimulating from. So just before we start going into the brain imaging finding, a lot of the data I'm going to show is based on the bold signal. So we are not really directly measuring brain activity, but we are indirectly measuring the blood flow response to neuronal activity, which actually is highly dependent on the ratio of deoxygenated to oxygenated hemoglobin. And it's also a response that is a bit delayed relative to neuronal activity. So what happens if you put somebody in the scanner and give them a painful stimulation? This is a meta-analysis of 420 studies. And basically, these are the most commonly seen areas, which actually reminds you of the cartoon I just showed you, which are actually activated in many, many salient stimulations in the environment. So there is nothing really specific about this map for pain, right? So you can see this in response to touch. You can see it in response to visual stimulation, auditory stimulations, etc. So acute pain in the scanner doesn't help us much. And actually, we learned very early that if we bring patients with back pain, for example, so clinical patients, into the scanner, and we give them acute painful stimulations here shown in gray, and we look at their own subjective pain rating of that stimulation, and we compare it to a group of healthy controls, you can see here in red that there's not much difference. This is the conjunction, and most of the areas are the same. And actually, if you contrast these maps, you don't see much differences between patients and control. So the paradigm of using acute pain in the scanner to study clinical pain is not really high yield. And so, of course, you can be smarter than just doing an on-off stimulation in the scanner only and do a general linear model. You can collect a large amount of data, like the group by Torwager et al. Here did, where they actually collected acute pain in the scanner, but they also collected response to warmth, response to social rejection, and response to friendly faces, I think, here. And they show that based on a large data set, you can build a model, and that model will distinguish between pain and all the other all the other modalities, if you want, that could give you similar activations, but their model actually does well in separating those other situations from acute pain. However, the problem with this, they dubbed it the neurologic pain signature, is that it wasn't really very helpful in distinguishing patients in chronic pain from pain-free control. But that's actually a much better way to approach, I would say, the understanding of how the brain basically processes pain. So what we did was first start by asking the patients to rate their own pain. And that's what we did a long time ago. We asked patients to actually report in the scanner while they are lying there, how their pain is fluctuating. And this is an example. Between 0 and 10, we asked them to rate their pain during scanning. And interestingly enough, it would fluctuate enough to help us do a linear model analysis. So this is one example. And if you do many of these in the scanner and average the maps that are related to pain, of course, and you remove a control from this, you get these areas. But what you see here is that the areas that really are related to spontaneous clinical pain, so there is no stimulus here, it's just the patient rating their own pain, are ventral striatum, amygdala, and the medial prefrontal cortex. So, and this is just here saying the same thing as here, that these activations are related to how much pain these patients had. And this was reproduced in several studies. So basically, when you ask patients how much pain they have, the areas that are tracking the pain are not sensory areas, are limbic areas. And then, like I said, these studies were reproducible. And then if you actually also look at brain connectivity between the medial prefrontal cortex and the ventral striatum, so it's a major, if you want, valuation hub in the brain. Connection means simply correlating two brain areas to see if they are going together or against each other or unrelated at all. So the correlation between the medial prefrontal cortex area here in red and the ventral striatum, which I showed you is related to motivation and liking and all of that, is tightly related also to how much pain they were reporting, these patients, on that day, the spontaneous pain. And this was also done by other groups where they looked at large-scale brain connectivity, and they found also that, you know, this connection of the medial prefrontal cortex to other areas is related to pain duration and also the severity of pain. So interestingly enough, maybe if we go back to healthy controls, although, like I said, it's not a good model to study healthy controls, but the healthy physiology of the brain gives us a hint of about maybe what is happening in the pain patient. So in this study, Tor Wager, again, group actually asked patients to modulate their own pain in the scanner. This was acute pain. And sorry, these were not patients. They were pain-free controls. So they asked patients, pain-free controls to modulate their pain up or down, right? And what they found was that, you know, the accumbens MPFC connectivity, which I just showed you is related to spontaneous pain in patients, is what was really mediating the relationship between regulating up or down and the patient's report. But this area didn't really change when they changed the intensity of the stimulation. So this area is basically mediating valuation, but it's not mediating sensory perception of the stimulus, the intensity of the stimulus, whether it was 47 degrees, whether it was 48 degrees, whether it was 49 degrees. And so this axis, if you want, is what becomes much more important. This valuation is what becomes much more important in pain patients. And so just another hint at these same areas, that these areas, the accumbens here, for example, is rich in opioid receptors. And when you look at opioid binding potential using PET in patients with fibromyalgia, with trigeminal neuralgia, and many others, you always find that these receptors are less available, which means that they are somehow more bound in the patients than in the controls. Whether that means that they have less receptors or that are available, or they have more internal opioids. We don't know, but we definitely know that there are disruptions in those circuitries. And this is just to show you that some groups actually try to reverse translate these findings into animal models. And in this study, they found that in animal models of neuropathic pain, you can actually modulate how much the animal would respond to a von Frey filament on the injured leg by modulating activity in the prelimbic prefrontal cortex or its projection to the accumbens, pretty much like what we see in humans. So the second conclusion basically is that brain activity in limbic areas, specifically the accumbens medial prefrontal cortex, seems to be tracking clinical pain intensity. So, but then we did something more, I would say, more helpful in understanding how these mechanisms develop. So we followed patients from acute to chronic pain. I'm gonna start showing you the functional changes, the functional data that we have. So what we did is we actually looked at subacute back pain patients in the scanner. We collected multimodal data of anatomy and function, resting state. And then we followed these patients again after 1 year. And you end up with some patients recovering and others persisting in having pain. And so we measured a lot of outcomes, but you can see that the recovered patients had at least 30% drop in their pain, but the persistent patients in red kept on having pain. So they became chronic. And this was consistent with other measures of pain, like the McGill pain questionnaire or the pain catastrophizing scale. But the first thing we noticed is that a smaller accumbens, again, in volume, predated the presence of chronic pain. So the patients who became later chronic had a smaller accumbens at baseline, and that volume, that decrease in volume persisted at follow-up. And this is what prompted us basically to look at the actual activity, what is happening in the accumbens. And so what we did is we extracted the brain activity from this area, and we looked at the frequency content. So you break down a signal from the time domain to the frequency domain to see how much of each frequency there is. And we noticed that in the chronic pain patients, you see a loss of the slow frequencies. It's called the slow 5 frequency. So you see a loss in the chronic back pain patients at baseline. Well, this was a cross-section here relative to the healthy controls, but not in the persistent patients or recovered patients at baseline. So these were retrospectively classified. So those patients who are subacute did not have this, if you want, marker of change in frequency in the accumbens. But then when we brought them back, you can see that the persistent patients start showing this marker, this chronic pain marker. And that was actually, if you plot the marker against pain intensity, you can see an inverse relationship. But the nice thing is that we could find the data online and validate these findings. So this is data combined from Chicago and Cambridge, showing actually that if we do the same contrast as here, you get the same loss of frequency in the nucleus accumbens, the same area I've been talking about. And also we tried with other data sets where we tried to classify patients, whether you classify persistent versus recovered patients or chronic versus healthy. And these of course, were at the chronic phase here. You can actually separate relatively well between groups. So it looks like we were up to a brain signature of chronic pain, and this was chronic back pain, and this was despite the fact that these cohorts were very different in age, in pain intensity, in pain duration, and even in reported depression. And so most recently, we've been trying to actually understand what does it mean? What does this frequency mean? What does it do? And one idea was to actually modulate the opioid receptor. So in this study, we actually gave back pain patients 30 milligrams of morphine, and we scanned them, of course, first, and then we gave them the morphine and we scanned them again. And we see actually after morphine, although the pain improves, this if you want, signature is accentuated, is intensified. There is even more loss of the slow 5 frequency in these patients. So despite the pain going down, the slow 5 frequency gets worse in back pain patients on an acute dose of morphine. And then here we are now trying to actually develop different ways of looking at the same areas by looking at the pattern of connectivity in those areas. You could think about this as a barcode for the areas. So and the barcode would tell you about the identity of each area, and then you can compare this barcode before and after an intervention. And we can see that again, the accumbens caudate changes a lot. This barcode changes a lot significantly after morphine. So that just tells us that that these areas are actually sensitive to a well-known addictive analgesics. And then when we looked at the anatomy, we also found that other areas of the limbic brain are actually predictive of outcome. So for example, a smaller amygdala will predict persistence of pain. A change in the shape of the hippocampus will also predict a retraction of the hippocampus will also predict persistence of pain. So basically areas involved again in memory and learning. And so this was actually what we found here in the amygdala and hippocampus was already reported by a study done in 2016, showing actually that a smaller amygdala and smaller hippocampus predict persistence of pain. And so also we wanted to look at other measures that we have collected in these patients, and this was more like exploration of white matter, what we can learn from the white matter. So briefly, you can basically get a map of water flow, if you want, of in the white matter in the major bundles, and thin out these major bundles into their centers, right? And you can look at these major highways in the brain, the white matter tracts, and you can look at how water flows in these areas and compare these groups I've been talking about. So what we found was that patients who are resilient to pain, shown in blue, they have increased fractional anisotropy. So there is a measure of water flow that was improved in their superior longitudinal fasciculus. This is a major, on the right side, this is a major attention pathway. It connects frontal and somatosensory areas. And it's important in navigation, body schema, etc. So on the left, it's important in language, but on the right, it's important in position in space. But the interesting part is that we had other data from other labs doing the same thing, and we were able to actually classify their own patients using just this area measure. I mean, this area's fractional anisotropy, so this measure of white matter flow. The interesting part is that also if you look at the recovered patients versus the persistent patients, you can see that the pain-free controls are somewhere in between. And that's because probably the pain-free controls are composed of both groups, people at high risk and people at low risk. And even more, actually, we got data from Meinheim in Germany, and we observed the same thing. We could also, again, in a third data set, distinguish between recovered and persistent patients by looking at this resilience marker. And the baseline fractional anisotropy in these patients predicted actually the change in severity. Of course, if you have more increased FA at baseline, you'll have more improvement in your pain. And this happened in the Meinheim data set and in the New Haven data set that we collected. So we don't know actually what this white matter data means, but we are trying to work on it now. We think that it is related to disrupted body schema. Like I said, from my reading of what this area does, is that maybe patients who have a better perception of their body, better representation of their physical body, if you want, in the brain, are the ones who are going to be resilient ultimately after an acute bout of back pain. But this is still just a theory. And actually, we started looking in the lab at measures of two-point discrimination and how well patients can actually distinguish body positions at baseline before they become chronic to see if that's going to be predictive based on the imaging data. So the third conclusion is that limbic brain areas basically predict persistence of clinical pain, whereas somatosensory attention areas predict resilience to clinical pain. Clinically speaking, in a nutshell, it might not just be in the back. That's what we are trying to say. And so now what we are trying to do is to see if those findings are generalizable, right? Can we reproduce those findings? Are they valid? Are they the same between chronic pain patients? So that's what we are trying to do. Now, I showed you an example with the diffusion data. It looks like with multiple small studies, you can reproduce that finding, but that's not a final verdict that these are highly reproducible. And of course, why do we need the biomarkers based on these mechanisms? Because we need to make rational decisions in the clinic, right? So until today, all we do is trial and error, especially in pain. There are no adequate analgesics in the clinic. And the way we choose an analgesic for chronic pain patients, I'm saying, is always by trial and error. And in the patients where we want to give an opioid, we worry so much now about addiction, and so we're not doing it anymore. So in one of the grants that we are working on now, actually, we are bringing together all these biomarkers I just mentioned and trying to put them together in a model to predict basically grouping. So will this model be the same for back pain, arthritis pain, trigeminal neuralgia, and depression? And back pain and arthritis pain, because this is like two musculoskeletal conditions. Back pain and trigeminal neuralgia, because trigeminal neuralgia is a typical neuropathic pain condition. And MDD, you would have figured out a depressed population because those areas that we have seen tracking chronic pain are highly important in affective disorders. They don't just track pain in life, they do everything else in life. And so one of our controls is actually to look at the brain of depressed patients. So some hint about reproducibility or not. So actually, this is a collaboration we have with the University of Würzburg, where they scan chronic and acute CRPS patients. And actually, if you compare those two groups using our biomarker, you can actually separate acute from chronic CRPS. So it looks like the chronic CRPS patients, which is a very unfortunate neuropathic pain condition, develop this loss of frequency in the accumbens over time. We are trying to look at arthritis. A preliminary analysis from data that we didn't collect, but that was available, actually shows that arthritis actually might be different. That we don't see the loss of frequency in the accumbens, but we see a gain of frequency in S2 insula. It's more like an acute pain. The arthritis pain is more like an acute pain. And this is what we are doing now with the diffusion data. So this measure, the FA measure, is a measure of water flow, but now there are better models actually to characterize the local white matter properties based on neurite direction and neurite density. And so if instead of using a simple tensor, we can use a three-component model to get more, if you want, fine-grained information about the local health of the white matter. And we're repeating basically the small study that I showed you now in a larger cohort to look at how these measures predict outcome. The good thing about this, which I forgot to mention, is that this is a scan that you can obtain in 10 minutes. So, and with time, it's gonna probably be shorter and cheaper. So this is something worth considering for translation. And the imaging analysis is not that complicated. Yeah, and we would like to, for example, in the future, predict treatment outcome, because the model, for example, now is what is that if you have back surgery, you have back pain, you do a spine scan, you find something like a lesion, and you correct it. That's the prevalent model now. But that can lead to a lot of problems because 40% of patients keep on complaining of back pain after the surgery. And we know that spine imaging findings do not predict outcome over and over again, because all the findings that we find in the spine are present in asymptomatic patients, and there are multiple studies showing that. And so what we want to do is use our models now to see if we can actually predict outcome of surgery, whether patients are going to be good candidates or bad candidates ultimately. Of course, also one could apply these measures to deciding which medication a patient should take, right? So they can improve our precision in treatment in the clinic. And that's it. Thank you.
All right, thank you, Paul. Very interesting. Now, does anyone have questions?
Okay.
Thanks, Paul. That was very, very useful to think about. Did I hear you right that patients lost hippocampal volume as chronicity developed?
Not exactly. Actually, what happens is that the patient at risk
has a lower
have a lower volume.
Okay.
That doesn't mean necessarily that chronic patients, if you compare them to pain-free controls, will show it.
Yeah.
Because there are two possibilities. Either the resilient patients have a larger hippocampus, right? Or the persistent are smaller than the recovered, but then the healthy could be in between, right? Because they have both types.
Okay, so I think that addresses my question, but I mean, are there any other correlations with the smaller hippocampus in terms of, cognition or anything like that? Or is that data out there? Yeah.
Yes, there is one data set at least showing actually that these changes we see in the hippocampus are related to the distortion of the pain memory. So in one study done in the Abkarian lab, they showed that actually patient tended to inflated the pain they reported last week, for example, and that inflation of pain was related to these areas in the hippocampus. Yeah, so in a sense, they could be related to the outcome. Yeah. John.
Okay. John?
Yes. Yes, exactly. Yeah, that's a great question. I didn't show the control. So what we used to do is we have them rate their pain and look at a bar on the screen. So they move the bar and tell us how much pain they have. And then what we do in the control, we take the time series of the rating, we feed it to the bar, and we have the bar move, and we have them follow the bar. So there was like a cognitive, visual, motor control, and that was our visual task, we called it. And then the maps that you see are a subtraction of pain from visual.
So that's great for getting rid of the sensory stimulation.
Yes. Yeah, we didn't do that, but we thought that rating the length of the bar is a kind of a magnitude estimation as well. But you're right. I mean, yeah, but they are following it, right? If it goes down, they go down. If it goes up, they go up. But it's not exactly rating an internal state. You're right. So maybe we could ask them to rate their hunger and see if we get the same thing or not. That was one idea we were discussing. It's a great question. Right.
Okay, we have some questions come in on Zoom. The first one is, as you know, anhedonia is also found in other populations, such as major depressive disorder and schizophrenia. Do you see a similar change in the nucleus accumbens volume in these populations? And what explains the change in volume?
So do we see the same changes? Yes and no, depending on the population. And sometimes the data is controversial. For example, in depression, there is data clearly showing actually that the accumbens is larger rather than smaller, although depression is the condition that where anhedonia is part of the major symptoms of DSM. And also a treatment study showed actually when you give patients with depression ketamine, the accumbens shrinks. Now, whether it's larger or smaller, what does it mean? That's the next question. We don't know exactly. There are lots of changes in the dendrites of medium spiny neurons. There are lots of changes in the dopaminergic transmission from animal models. What we hope to do actually is to actually scan animals with chronic pain models. And now here, for example, in our new 9.4 Tesla, and actually connect the two modalities, the images with actual tissue studies to really know what does it mean in different conditions. I don't know of any other condition where accumbens is shrinking, but I think in schizophrenia, probably it's also shrinking. So that raises the question of definitely of specificity. Yeah.
Okay. Well, we have more questions that have come in on Zoom. One is, you described changes in chronic low back pain and imaging after acute morphine that showed some abnormal signaling.
Yes.
Is this consistent with other opioids, such as buprenorphine?
That's actually never been done. And this is what we were looking into doing now, because there are lots of patients who are on high-dose morphine or morphine-like drugs that are being switched to buprenorphine. No one has ever looked at buprenorphine in chronic pain patients in the scanner. No one. There is one study done on acute pain and pain-free controls. And there are PET studies in the context of opioid use disorder. I would imagine that actually buprenorphine would do better in the sense of not making things worse in that sense, while inducing the same amount of analgesia. As you can see that we see an accentuation of this marker, but there's a drop in pain. So it means that that marker means something else in addition to the pain intensity. It means the disease itself, so at least. So we think that buprenorphine would actually improve things relative to morphine.
Okay, well, while the people present, we do have a question live. We're going to get you a microphone.
So pardon me if I bungle some of the vocabulary here, but I was recently reading an article in Translational Science out of University of Wisconsin, looking at similar things, but from a different angle. The idea of examining some of the functional imaging of folks who are considered expert mindfulness meditators. And noticing that the greater tolerance of pain is associated with increased capacity for attention pathways. And so I'm wondering if you could think a little bit about that kind of feedback, which seems to map onto some of the things that you were talking about as well, and speculate on therapeutic interventions for folks with chronic pain in this neuroscience perspective.
Yeah, so mindfulness meditation or people who do it well it's a very well now validated treatment for chronic pain. There are many clinical trials showing that it actually works. Now, whether it maps to the same type of circuitry I'm talking about, it's yes and no, because people who have done the mindfulness meditation studies, unfortunately, I would say it looked at actually patients in the scanner, but in response to acute pain. They didn't look at their own spontaneous pain. And I'm not aware of resting state data, so no stimulation at all. So, but yes, it works, and it actually could map to our areas. Whether it has been done, I would not quite, no, not yet.
Okay, well, another question from Zoom. Do you have any thoughts on the hypothesis that physical pain and social pain share neural substrates? I recall there being controversy over Eisenberger's findings, but I'm not up to date on it. Great presentation.
Whether they share neural substrates?
Yes, physical pain and social pain.
I don't know actually the answer to this question quite well. What I can tell you is that if you notice the data that was published by Torwager, they used actually social rejection, which, if you want, a kind of social pain, as one of their controls. And although social pain and acute pain, if you do them in the scanner, you see the same areas involved, the pattern of activity and the weights of each area is different. Now, how does this really differ between acute pain and social rejection, or social pain, I would say? What are the specifics of the circuitries? Which areas are much more important than others? I'm not quite sure yet.
Yeah, that would be a really interesting grant proposal.
Yes.
Okay, well, a couple other questions. Do persons with Huntington's disease show altered pain perception given their pathology that involves key areas?
I don't know about Huntington. I'm not sure, actually. That's a great question. But I know about Parkinson's disease, right? Parkinson's disease, number one complaint is pain. But Huntington, I have never really looked. It actually would be an interesting question to see with the degeneration of the caudate, whether their pain, whether they start developing actually pain or not. Yeah, but that's a good question.
Yeah, you know, we have a well-established Huntington's disease program here, so that's something that could be looked at.
Yes.
Okay, last question. Do you see a connection between the somatosensory smudging you described and somatic experiencing approaches used in psychotherapy for pain, such as body scan, pendulation, etc.? We often see benefits when we're able to help tune in more to their bodies as a whole, not just painful areas.
Exactly. Yeah, I think this is what must be happening maybe in these types of treatment, where you would help patient not just recover the or improve their body schema of the back only, but of everything in their physical world and how they navigate this in space. This is the hunch from the data that we get, because this right superior longitudinal fasciculus is new to us, and we are trying to explain why it predicts resilience. I would think that's the case, and this is what we are trying to work on in the lab, to collect some measures of body perception and to see if it's going to relate to resilience and to the brain area.
Okay.
Brain bundle, sorry.
And I guess I'll take the last question.
Okay.
We have a couple of minutes left. One of the slides earlier in your talk with the experiment with the pudding.
Yes.
It seemed like the pain group was different than the control group on the food, but not on the drinks.
Yes. Yes.
Not just sugar, but even on the juice. So why would that be?
So what happened there is that on the sucrose solution, there was no difference. Yeah, there was only difference in the pudding data, right? You're asking why is it fluid versus solid?
Yeah, because I thought in addition to the sucrose, which I assume was mostly water with sucrose.
Yes.
The graph above it said juice, and it didn't look like there was a difference there either.
Right, exactly.
So it's not just about food.
Yeah, I mean, I don't know why it's solid food versus sucrose solution, the difference. Now, we repeated this study, but instead of giving patients sucrose solution, because we thought about exactly that question, we converted it to Jello, right? So we gave patients Jello and pudding, right? One was sugary, one was fatty. And we observed almost the same thing as in this experiment. So it might not be the consistency of the food. The theory why it's pudding is that I didn't go into it, but there is clear evidence from animal work, and this is what prompted me actually to try the fat, is that if you modulate the opioid receptor specifically in the accumbens, you change completely the preference of the animal when it comes to sugar versus fat. For example, if you inject opioids in the accumbens and present a well-fed animal with chow versus fat, they will only go for fat, like chocolate or something. They will never touch the chow or even the sugar. If you give them just sugar, they will only go for the fat. So we thought that there is a clear intersection between disruption of opioid receptor in this area of the brain, the accumbens, and the perception of food, right? The perception of fat. And it's all, I think, in a sense, we can put all of this together, that when you have chronic pain, it's something that you are always factoring into your decision making. And the decision making, evolutionarily speaking, is all about basically surviving and finding your food. So
Okay. Just as a final comment, I guess, there was a few years ago, John Crystal, your former chair,
yeah,
in an article on computational psychiatry, made a comment that despite all the research in psychiatry, we still don't know the biological basis of even one symptom of one psychiatric disorder. And I think what's interesting about your work is it really is narrowing in on the experience of pain, which you could say is a physical problem, but clearly it has so many psychological aspects to it. And so it's a really good example, I think, of how to approach, studying the biological basis of a subjective experience. So anyway, congratulations, and thanks for another great presentation.
thank you. Thank
Thank you all for coming, and please fill out the evaluation.